Presentation. Okay. Duh. I need to open up the presentation. So while this is loading, let me just say, it's loading now. I think yesterday I finished grading the practicals, and not this one. And then today I caught up on any other outstanding graded assignments. So I'm up to date on grading if you've been waiting to see that. So we were on this stuff.
Yes.
Okay. Had I explained what exogenous and endogenous presentation or so in the previous stuff where I was talking about the maturation, the positive selection test, if you will, for T cells is testing that they interact with this molecule called MHC. Now, MHC stands for something, the name of which would just confuse things because it's not exactly what it does. It was named before when it was first discovered and named, it was based on the function that it was discovered for, and it's responsible for a number of other functions. So don't worry about what it actually stands for. We'll just refer to it as MHC. It comes in two types, class 1 and class 2 MHC. Phagocytic cells will process antigens, ingest and digest microbes or something like that, and then present parts of that on its surface with this molecule called MHC. And phagocytes use class 2 MHC. Cells in your body are continuously presenting antigens on their surface. Most of those are antigens that we would consider self-antigens. And at this point, based on the maturation process that I discussed before, we can kind of ignore self-antigens because we should have eliminated any lymphocytes that would recognize self-antigens. But when cells in your body are doing something abnormal, if they're infected or they're showing signs of disease like cancer or something like that, or they just start malfunctioning, they'll present antigens on their surface that are incorrect. And body cells, non-phagocytic cells, use class 1 MHC. The difference between class 1 and class 2 is just what type of T cell interacts with it. Oops. Okay, I've already read this. Endogenous presentation, meaning presenting antigens from within your body, are presented by infected or damaged body cells to what are called CD8 cells. Those are, you can't quite see the subscript there, but those are cytotoxic T cells, T subscript C cells. It requires class 1 MHC. And then exogenous presentation Actually, I keep forgetting I'm recording this stuff. And what I write on the board doesn't really help for, like, Casey, who's homesick. She emailed me today and I said, wow, you're missing the immunity stuff, but you understand that you're sick and shouldn't come into class. So more to you. Okay, so let me actually write this down real quick. So I'm just going to rewrite what I have up on the board, hopefully a bit more legibly. So endogenous presentation to maybe it this way, is infected or damaged body cells to CD8 or DC cells. Now I can't put a subscript on the board here. The thing that I'm using just doesn't allow that. So I just put a lowercase c there instead of the subscript. And it requires class 1 MHC. I was hoping it would fit. And then let me just copy this. Start doing all that typing all over then. Exogenous presentation is from phagocytes to what are called CD4 cells. Those are helper T cells, so the subscript's an H, and it requires class 2 MHC. Okay. So that's just writing it down. I have a couple of pictures that I'll show you in a second that illustrates that. So, the T cells here, they're the ones that are responsible for the cell-mediated immunity. And so they have to interact with other cells. And MHC is essentially how cells interact with each other, specifically how other cells interact with T cells. And depending on where the antigen is coming from, endogenous antigens are coming from cells that are already in your body. The ones we're most interested in are the infected or damaged ones. Normal body cells would present also, but there shouldn't be any T cells for them to present to because we should have eliminated any T cells that would recognize normal self-antigens during maturation. And they use this class 1 MHC. And then exogenous antigens are presented by phagocytes that have processed them to the CD4 cells or helper T cells, and they use class 2 MHC. So let me go back to the presentation to pick up with that idea. Yeah. Now I showed you, we established I actually showed you a different picture originally, but it's the same kind of thing. So phagocytes that are presenting exogenous antigens, like a microbe, they ingest that, break it down, and then what's left over is going to be available to be presented. So this sort of picks up with sort of what the phagocytic pathway left off, or the breakdown products of that, and they can be used. We don't have a picture showing a body cell at this point, but we'll see that in a second. But at this point, I'm just reminding you of the phagocytic stuff. For the sake of the pictures I have to be using in this presentation, I'll include this, which just introduces what these class 1 and class 2 MHCs look like. So in the little inset at the top of both of these, there's a cell membrane represented by this sort of double line yellowish looking thing. And the MHC is a protein that's on the surface of the cell. This is the outside of the cell, and it has in it a piece of that antigen that's being presented. And for the source that I'm using in these particular pictures that I'm showing you, the MHC proteins are depicted this way. The one with the little circle in it like this is class 1 MHC, and then class 2 MHC is just sort of this bilaterally symmetrical cartoon here. So they're similar kind of things, they just look slightly different to represent that they're different versions of them. Class 1 MHC is presented to CD8 cells, which when they're fully mature are mostly going to be what we call cytotoxic T cells. That's T with a subscript C in what I wrote before. And these are the ones that we usually think of as the attack cells. That was the term that was used in the podcast we listened to. They can also give rise to memory cytotoxic T cells, which are available for future infections by the same thing. And then they also give rise, CD8 cells also give rise to a special type of T cell that we won't talk about any other time except for here, called suppressor T cells. The name of these is actually kind of in transition. When I originally learned about the immune system, they were called suppressor T cells. Nowadays, they're more often referred to as regulatory T cells. The subscript after the T in their abbreviation would either be T subscript SUP for suppressor or T subscript REG for regulating. But these are the lullaby cells that are mentioned in the podcast. So they suppress or regulate the activity of other immune system cells. And in the hygiene hypothesis, what they're kind of getting at is that these cells are not as active, or there might not be as many of them around because that the term that was used in the podcast was positive regulatory advantage, I think. That advantage of having a low-level infection of something like a hookworm is lost, and so these types of cells aren't produced in as the same kind of number that they should be to keep everything in check. So class 2 MHC interacts with what are called CD4 cells, which the mostly become what we call helper T cells, but also give rise to memory helper T cells that are available for future responses to the same infection. So this picture, there's not exactly a comparable picture in our book. In the book, it just, in the text, explains briefly what these types of cells are, but they don't have a picture like this. And then these two pictures, again, from this other source that I've been using for this presentation, just show how the two types of antigen-presenting cells present those antigens. On the bottom here, we have a phagocyte, so it's ingested a microbe, which then gets digested by the contents of the lysosome, and then the bits and pieces that are left over, that residual body that was in the picture I showed you a little bit ago, get presented on the outside surface by class 2 MHC. A vesicle from the Golgi apparatus will bring one of those MHC molecules over. It'll merge with the residual body, and then this will merge with the cell membrane, and whatever's in there that's not attached will be just released as waste, so to speak. But then the MHC, class 2 MHC protein, will be in the surface of the cell, presenting that antigen, a little red dot as it's represented here, to the CD4 cells. So this is what's happening in phagocytic cells. This is what's happening in regular body cells. Now, in regular body cells, the self-antigens, which we're going to kind of ignore at this point because they shouldn't be an issue, thanks to the maturation process we talked about. Self-antigens are going to be made by cells in the rough endoplasmic reticulum, and those will be processed by the Golgi apparatus. Transport vesicles will put membrane-bound proteins on the surface of the cell, or secrete things like hormones out of the cell or whatever. Or collagen that's being secreted by fibroblasts or whatever will be secreted that way. Any proteins that the cell makes that it keeps inside the cell won't use the endoplasmic reticulum and Golgi apparatus. They'll just be made by free ribosomes in the cytoplasm, and they'll escape this process. But proteins that are made in the endoplasmic reticulum will end up being presented on the surface of the cell by class 1 MHC. Now in this picture, which they've labeled infected cell, they're suggesting that either a viral infection or a microbial infection getting into the cell will introduce antigens to the cell, or I should say, will cause the cell to make antigens based on its being infected, like making new virus particles. And then those things that are being made by the cell will be presented on the surface, just like self-antigens, but these signs of infection are abnormal and your immune system should react to those. So that's how the cells present antigens on their surface. The bottom picture is presenting exogenous antigens through phagocytes and class 2 MHC. The top picture is presenting endogenous antigens on the surface of body cells through class 1 MHC. The picture is highlighting specifically infected cells, but if it's a cell that's cancerous, for instance, then it's going to show signs of having that kind of disease. For example, colon cancer has a marker associated with it called CEA. That stands for carcinoembryonic antigen. The carcino part of that word is referring to the fact that it's found in cancerous cells. The embryonic part of that word is telling you that it's a protein that's normally made in the embryo. So what it is this cancerous cell has turned on a gene that's usually turned off after embryonic development. And that's what cancerous cells tend to do. They activate genes by accident sometimes. A lot of the genes that get activated help the cell grow out of control and form a tumor, but sometimes there are other things that are activated. And in colon cancer, this particular thing is used as a marker for that cancer, which is very helpful clinically. I don't know what it does for the cancer cells itself, but a cancerous cell in the colon will produce this normally embryonic protein and present it on the surface of a cell. Now, your immune system learns about self-antigens after birth. So embryonic proteins would not ever be considered self-antigens because they're not present in the mature postnatal to adult body. But in cancer, that kind of thing can be presented. So that'll be made in the cancerous cell and presented on its surface and marked as being a diseased cell also. So there are other things besides infection that can present antigens on the surface of body cells, but this particular picture is sticking with the infection part of it. Yes?
So how would the virus present as an endogenous?
So we consider virus particles that get into your body from outside as exogenous. But once they've infected a cell and forced that cell to make new virus particles, the new virus particles are endogenous because they're being made by the cell that's being infected. So yeah, that's a little bit of an odd example, but it's because that cell that's infected is making new virus particles. And endogenous just means made in your body. So the new virus particles are being made in your body, so they're counted that way. And an infected cell would present those.
Class 1. Class 1.
Right, with class 1. The mature cells that I described last time, T cells mature in the thymus, B cells B cells mature in the bone marrow. Those two locations are considered primary lymphoid structures, organs, tissue, whatever. All of the other structures in the lymphatic system are considered secondary structures because it's not where those cells mature. And after they have matured, this B cells and T cells will then move out into the secondary structures like lymph nodes, the spleen, tonsils, mucosa-associated lymph tissue, all that stuff that we talked about that was not primary tissue is where the mature cells go. Now, they're also considered naive at that point. Okay, so they've gone to school, they've learned all they need to learn to be immune system cells, but they haven't gotten their first job yet. So they're out looking for a job. And hanging out in a lymph node or whatever, they're waiting until they run across an example of the antigen that they're specifically targeting. They aren't necessarily ever going to see that antigen. But if you are ever infected with something or exposed to something from outside your body, then there should be a lymphocyte waiting to interact with that. For example, if you have type A blood, then you're going to have lymphocytes that make antibodies against the type B antigen. You're type A blood, so you don't have the type B antigen in your body. So you're still going to make antibodies against that. It's highly unlikely that you would be exposed to type B blood. Most of the time when somebody's exposed to blood from outside their body in a way that their immune system is going to react to it, it's because they're getting a transfusion. And transfusions ideally should pay attention to what your blood type is. So if your blood type's A, you should not be getting a transfusion of B type blood. So it's highly unlikely that the lymphocytes that you have in your body that are specifically going to recognize type B blood are ever going to interact with that particular antigen, but it's possible. More likely you're going to have lymphocytes that are specific to, say, the influenza virus or something like that. And you are probably going to be exposed to the influenza virus. So when you are exposed to the influenza virus, the lymphocytes that are hanging out in the secondary lymphatic tissue get activated. So that's sort of when they move from being mature but naive to being actual components of functional immunity. So these panels all show the process of activating the different lymphocytes. We're going to start with the CD4 cells, which become mainly helper T cells. Now, the antigen presenting cell that's shown here is a phagocyte. And so it's ingested, let's stick with the influenza virus example. It's ingested influenza virus that you've been exposed to and processed that, and it's showing part of its, the viral particle that's been broken down, it's showing that on its surface with class 2 MHC. Now it's going to expose that, or present that, I should say, to the CD4 cell, and the CD4 cell has on its surface what's called a T cell receptor. I haven't talked about T cell receptors yet. Okay. So far, when I've been talking about the molecules that specifically recognize antigens, I've been saying antibodies. Antibodies are made by B cells, and they are secreted into the plasma. They're a type of plasma protein. T cells don't make antibodies, instead they make T cell receptors. The two things are very, very similar. I mean, they're different proteins, but they're similar in that they have antigen binding sites on them that will specifically recognize the three-dimensional shape of molecules. Let me get out of the presentation for a second just to talk about what this is. Let's see here. And the easiest way to do that, I'm just going to do an image search for. Nope, that's not what I want. Antigen binding site. So I just did a search on Google for antigen binding site, and I'm getting a whole bunch of images representing this. Antibodies, that's not a very big picture. That'll do. No, actually, that's not the one I want. Okay, this will do. So antibodies are usually represented in sort of a Y-shaped structure schematically. They're made up of 4 polypeptide chains. 2 of them are long chains, which kind of give that Y shape to the molecule. And 2 of them are short chains, which double up in the sort of arm part of the Y. Now, when you're talking about protein chemistry, you don't call proteins long and short. Instead, you use the terms heavy and light. A long protein is a heavy protein, a short protein is a light protein. So we have heavy chains that make up sort of the body or stem of the Y and continue up into the arms. And then the light chain pairs up on either side with the heavy chain, completing that Y structure. The proteins or polypeptides that we're talking about here, these chains, are structurally kind of separated up into a bunch of different little segments or loops, or there's another word that's used later on when we look at this, which are just depicted by the different colors here. The half of the light chain and the quarter of the heavy chain that line up, making the outermost part of the Y. Like if I'm standing here and I'm a antibody, then the ends of my arms or the hands are attached. That part out there is what we call a variable region. And then the other half of the light chain and the other 3/4 of the heavy chain are made up of loops that are called constant regions. What that's referring to is that the gene for that chain has a variable region that when the lymphocyte is first produced in the red bone marrow, the DNA randomly resorts itself to make a unique gene that'll make a unique antibody. And then the constant regions don't change, so that the rest of the antibody has a pretty standard structure to it. The variable region gives rise to what's often depicted as sort of a puzzle piece that fits in with antigens. So the variable region in either piece of the Y-shaped part of the antibody here gives a 3-dimensional shape at the end of the antibody that fits perfectly with the particular shape of an antigen. So in this picture, what they're showing you is this particular antigen that's kind of a mustard yellow color, I guess, is the one that matches the antigen binding site on this antibody. And the other 5, which, because I'm buying so many diapers, just look like icons from a diaper box these days to me. Anybody? Okay. But so those other 5 shapes here do not fit into this antigen binding site. Okay. This depiction here is a little less symmetrical. The antigen or the variable region in the heavy chain, the variable region, and the light chain are not necessarily going to be identical or mirror images to each other. But together, they sort of define a shape that a puzzle piece kind of reaction can take place. So only something with this particular shape, this antigen, will fit into that. Other shapes won't. Now, the two antigen binding sites on an antibody are identical because the light chain is just one light chain that's duplicated for the two sides, and the heavy chain is just duplicated to make the two sides there. So this antigen binding site and this antigen binding site are identical. They bind to the exact same antigen. Okay. This picture shows in a little different kind of depiction of what these things look like, sort of the variability in what the antigen binding sites and their complementary antigens might look like. So here we have kind of little nubbin sticking off of something that fits specifically to that. Here, this can sort of latch on to part of the protein structure of something. This is complementary to that shape. This is the first representation I've seen in this kind of cartoon way of part of the antibody sort of sticking out and fitting into the antigen. But whatever it is, there's a very specific match between the antigen and the antigen binding site on an antibody. This picture shows a T cell receptor, which has a variable region. Okay, first off, instead of a light chain and a heavy chain, it has two chains that are referred to as alpha and beta, which are roughly the same size. At the end of either chain, there's a variable region, just like in the antibodies, and that's what establishes the shape of the antigen binding site. And then there's a constant region, which in this particular picture depicts sort of the rest of the thing. The depiction in the book has a constant region, and then the rest of it's just what goes through the cell membrane of the cell. But however you want to think about it, there's a variable region at the end, and then the rest of it's all pretty standard to give the shape of the thing. Now, there's only one antigen binding site on the surface, in the T cell receptor. There's two in an antibody, and that's because antibodies can actually bind to two separate antigens and link them together, like you see here. So these two are linked together, but then this one's also linked to a third by another antibody. T cells don't do that sort of thing. And when we get to talking about B cells and antibodies, we'll touch on why it works that way. But that's a T cell receptor. Okay, so I just wanted to jump out for a second to explain that to you because T cell receptors are important here. So the CD4 cell, which is a type of T cell, has on its surface a T cell receptor, which is going to recognize the antigen that's presented by the antigen presenting cell. It also has a protein called CD4, which is why it's called a CD4 cell. CD4 recognizes class 2 MHC. So when the T cell receptor recognizes its particular antigen and CD4 recognizes class 2 MHC, those two binding events or recognition events, whatever you want to call them, will cause the antigen presenting cell to release a chemical signal called co-stimulation to the CD4 cell, which will cause it to become activated, and the activated cell proliferates, meaning that it makes a bunch of copies of itself. And all of those copies have the exact same gene for its T cell receptor, so every T cell receptor made by all the copies of the activated cell recognize the exact same antigen. And they differentiate into active helper T cells or memory helper T cells. The memory helper T cells are inactive for this particular infection, but if another infection of the same type comes along, then they're ready to help fight off the infection. The active helper T cells release chemical signals that belong to the class called cytokines. Cytokine is just a very broad class of a hormone kind of chemical that's released. And I say hormone kind of because cytokines released from white blood cells sometimes interact with neighboring cells in a paracrine effect, or sometimes even autocrine, they'll affect themselves. And they don't necessarily have to travel through the bloodstream, but they're still signaling molecules like a hormone. So those are helper T cells. To understand what they do with the cytokines, we have to understand the other types of cells. So the next type to talk about is the other type of T cell, which is called a CD8 cell. And the antigen presenting cell that's going to present to that will be a body cell, and in this case, again, they're saying infected, but it could just as easily be a diseased or damaged cell, like something that's cancerous or a cell that's malfunctioning of some sort. And it's going to present antigens on its surface using class 1 MHC. If we look a little closer at this insert, we see the T cell receptor for the T cell, which is recognizing the antigen presented by the antigen presenting cell. And then the CD8 cell is called that because it has on its surface a protein called CD8. CD8 recognizes class 1 MHC. So again, the T cell receptor antigen binding and the CD8 MHC recognition, those two things have to happen. And a signal is released by the antigen presenting cell that co-stimulates and activates the CD8 cell. Now, this depiction doesn't talk about it here, and it might be a kind of rare occasion, but sometimes the presenting cell in this situation is so far gone that it can't co-stimulate the CD8 cell. So if it's infected, that might interfere with the ability of the cell to do what it's supposed to do, and it might not be able to release the co-stimulating factor directly. And that's what helper T cells are for. They help activate other lymphocytes. So if the CD8 cell can't get the signal directly from the antigen presenting cell, the cytokines that are released from the helper T cell can do the job for this if it's needed. When they are activated, CD8 cells proliferate and differentiate again, making many copies of themselves. That are identical in that they all have the exact same T cell receptor, and they differentiate into either the active cytotoxic T cells or memory cytotoxic T cells to be available for future infections. Sometimes active cytotoxic T cells are also called killer T cells. Basically the same thing. Cytotoxic means toxic to cells, so they're going to kill other cells. So sometimes we just call them killer T cells. When they're active, they leave the lymphatic system and go out into your tissues and find other infected cells, which are infected by the same thing, so that their T cell receptor recognizes the same antigen that they were originally activated with. And they have CD8 on their surface, which recognizes class 1 MHC, which means that they're finding an infected body cell, not a phagocyte that is presenting the antigen. And then they'll kill the cell. They can do that by releasing cytokines themselves, which can stimulate apoptosis, basically saying to the cell, you're infected, die. Or they can release toxic molecules, just generally called lymphotoxins, which will disrupt the metabolism of the cell, which hopefully will cause the cell to go into apoptosis also. I think there are lymphotoxins that will break down DNA, and there's sort of a built-in signal in any cell that if the DNA starts to break down, it automatically becomes apoptotic. So there could be that kind of thing there. If those sorts of things don't work, if the infected cell is so infected that it won't go through apoptosis, then the killer T cell can release something like perforin, which is a protein that perforates the cell membrane. That's why it's called perforin. And that destroys the plasma membrane and causes cell lysis. In this picture at the top, they show the killer T cell finding the infected cell, and it says lysed cell. Lysis is not the ideal situation here. Really, you want to see apoptosis occur, okay? Because that's easy for white blood cells, phagocytes to get in and clean up after. lysis will release whatever's inside the cell and cause problems. So if this cell is infected with viruses and a killer T cell comes along and can't cause apoptosis, it causes lysis, then it's going to release any of the viruses that are inside the cell to then go out and potentially infect other cells. So it's not ideal to just lyse the cell. It's actually better to cause apoptosis, so that whatever's infecting the cell remains inside the apoptotic vesicles, and the phagocytes can clear it away from your system. Now, there's a nice movie that I want to show you that illustrates this a little bit. In the video content part of Blackboard, where I have YouTube videos that I've found that apply to most everything in the course. There's some I want to show you. So it starts with the autonomic nervous system, which is one of the first things we started the semester with. It goes on into the endocrine system and the cardiovascular system. And then there's several that I found about the immune system if you're interested in watching them. The first one there with the measles, that's the ABCs of epidemiology. It's just kind of a cute video. This one, if you have time, I recommend you watching it. This is a neutrophil, or a phagocyte, that's filmed chasing down and engulfing a little bacterium. So this sort of double dot, black dot looking thing, that's a bacterium, and this cell sort of chases it through a petri dish until it grabs it and ingests it. So you're welcome to watch that. It's like 30 seconds, 29 seconds. There's no sound on it. I actually feel like there needs to be a player piano in the background, like a silent movie from the 20s or something like that. A few others that I found among the years. Then what I want to show you today actually is not this video right here, but rather a video that is at this link. When I'm putting videos in here, it has to be a publicly available video on YouTube for me to embed it into Blackboard. And what I want to show you is actually not a publicly available YouTube video. Instead, it's a YouTube video that the makers embedded into another page. This video here is actually by the same people and some of the same footage from what I'm seeing. I think it was just the first video they made of this material, and then they got better videos and they put it together and put a voiceover and put it up on this LabRoots website. So let me go to this website. If you want to, you can watch this video directly, but anything it shows is actually visible here. So there's a little bit of title slides here at the beginning, but eventually there will be a voiceover that talks about it, and I gotta get rid of that. Now, before you get started, let me just say, and you kind of saw from the frame before I hit play, these cells are visualized using fluorescent markers and viewed under fluorescent microscopes, which is visually very stunning. It's really nice. And they highlight different types of cells in different colors. And in fact, there's a part where they show a particular thing inside of a cell using fluorescent colors to light it up. These are cytotoxic T cells that have been activated specifically against a marker for cancer. They don't say what type of cancer it is necessarily. But they're basically hunting down cancerous cells and destroying them. I think a lot of this is done in vitro, which means in a petri dish or something like that. And they're not filming real killer T cells out in your body doing something. But this is how it would basically work.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explode the surface of the target. Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate. Now we'll leave you with a few more movies of T cells doing what they do best, killing cancer.
Okay, so you can watch the rest of that if you want to see more carnage, but it doesn't show anything new for the rest of the video. I like this video because it shows a white blood cell, specifically a killer T cell or cytotoxic T cell, moving. And as he describes it, it pushes its cell membrane out and pulls the cell along behind it, which is essentially how an amoeba moves also. In this, it looks like it's just moving across a black background, but probably there are a bunch of other cells that it's crawling over. They just haven't been marked with a fluorescent dye, so they're not visible. And then in the next section, they've marked a cancerous cell with a blue dye, and then the killer T cell has the red color to it. And when a killer T cell finds the cell that is presenting an antigen that it recognizes, it'll start to reach out all over the surface of the cell to find more antigens and recognize more definitively that's the cell that it wants. And in the next little section, they talk about the lytic granules, which they're staining specifically red inside the cell. They start off in the back of the cell, and when it finds its target, those lytic granules will move forward, and that's what gets injected into the target cell to cause it to die. So they do that sequence twice in a row. And then they move to this picture, which shows 3 cells that have all been stained to show you the lytic granules. And then this is just really kind of cool microscopy that they isolate this one cell and they rotate it so you can see sort of the 3-dimensional structure of the cell. And what's happening is these little lytic granules are moving towards the surface that the T cell is pressing against the target cell, and those granules are going to release those things into the cell. And then the last sequence is just showing you several examples of when these T cells find a target cell, they latch onto it, and they inject their poisons or whatever into it. And they've put a special chemical inside the cell that when that stuff is released into the cell, it causes the cell to flash red. And some of the cells, they flash once and then they flash a second time because they're getting sort of multiple doses of this. It always, to me, kind of looks like if you've ever seen like an old World War II movie or something like that, a soldier will rush up to a tank and lift up the hatch on the top of the tank and throw a grenade inside and close it, and then the grenade explodes inside the tank. Excuse me if that's a little overly graphic for you, but that's kind of what I picture when the cells flash red. That stuff's been shot into the cell that's going to cause it to die. So again, if you want to watch that video yourself in Blackboard in the video content part, this is the embedded video, which is by the same lab. It's some of the same footage and some older footage that they have. But the lab roots one is one I just showed you. There's a link underneath the video that has that voiceover and explains what's happening there. Okay, so if you want to go back and look at that yourself. Now, before I move back to the presentation, let me move over in the book. Oops, too far. The section on T cells. First off, they explain the structure of the T cell receptor, which I did a little bit ago. This is a weird picture, but it's just showing T cell receptors that are all recognizing an antigen. What's weird about this picture is that T cells recognize cellular antigens. And so this has 3 cells all recognizing a molecular antigen, which is not what T cells do. But for the sake of fitting it all into a picture, I guess that's why they decided to do it that way. In the book, when they're talking about antigen-presenting cells, this is what they use to demonstrate what's going on. Now, for sort of editorial reasons, the book decided to do this, and I kind of understand, but it was a slightly confusing choice. This cell, which they have several pictures of this type of cell in this chapter, is a dendritic cell. It's not a white blood cell. It's probably related to white blood cells, but it's a cell from like the skin or a mucous membrane. But they're also phagocytic. The interesting thing about dendritic cells, which are actually fairly rare compared to some of the other cells we're talking about, but it's the one that this book shows, is that they're able to present antigens on their surface from either endogenous or exogenous sources. And so in this picture, this is how our book describes those two things. It's showing exogenous presentation in the sense of a pathogen being ingested by the cell, digested by the lysosome, and then MHC class 2 is introduced and it binds to that antigen, which is then going to be on the surface of the cell. Over here, and this is the really weird thing about dendritic cells, this is not showing an endogenous antigen that this cell made, but rather one from another cell that's been infected. So dendritic cells are activated when, say, influenza virus infects the respiratory mucosa. So those are endogenous antigens that are made by the respiratory mucosa, and the dendritic cell is presenting, which is really weird. It doesn't fit with what I've told you, but that's what happens. So instead of these being antigens made by this cell, these are antigens made by another cell in the body that the dendritic cell gets. And then this is MHC, class 1 MHC, and that presents out on the surface. Another annoying thing about this book is they did not graphically show a difference between class 1 MHC and class 2 MHC. The pictures I've been showing you. So out here, the exogenous and endogenous presentation pathways that are shown converge, and they just say, and they're shown on the outside of the cell. But it doesn't tell you whether this is class 1 or class 2 MHC. And then they're going to interact with a T cell, and the CD protein on the T cell will recognize the MHC. So whichever one the dendritic cell is presenting will match up with the correct T cell because the CD protein, CD4 will recognize class 2 MHC and CD8 will recognize class 1 MHC. That can activate cytotoxic T cells to go out and find the cells that are actually infected in your nasal mucosa or whatever. Respiratory mucosa, I think is how I said it before. It's just the dendritic cell is coming from that location saying, here's evidence of some of the cells in my location that have been infected. It's a little bit of a strange twist to the story, but it's really not any different from what I've talked about. Just special cell that does that rather than the body cell itself. Okay. So I just want to point that out about how this book does that. And then also what I just showed you about activation, our book shows it this way, which isn't different from what I showed you, it just puts it all together. Oh, I take it back. This is the picture for maturation, which is happening in the bone marrow or the thymus. I don't think that's what I want to show you. These are the pictures that show activation, like I just showed you before. So this is our book's depiction of the cytotoxic T cell activation, which I showed you a second ago. And all I want to point out to you is that in this book, again, they're using dendritic cells as sort of the antigen presenting cell, which is kind of an unusual choice, but it's what they did. But still, we have class 1 MHC presented to a CD8 cell or cytotoxic T cell. And that's going to cause proliferation and differentiation of the cells, giving us active cytotoxic T cells, which are going out to destroy the infected cells, or memory T cells that are going to be available for future infections. This depiction of lysis of the cells, as well as the one that I was showing you in the presentation, both, again, are not ideal. You would rather see the cells destroyed by apoptosis than lysis, but the pictures always show that. Also, I want to point out the movie I just showed you is real cells really doing what they're supposed to do. And in a textbook, they're always going to show a gap between the two cells where they can enlarge the molecules involved. A T cell receptor is nowhere near that big relative to a T cell. And MHC is nowhere near that big relative to the antigen presenting cell. This gap here, or the gap here, we just saw isn't real. The T cell kind of latches on and wraps around at least part of the target cell. And this is happening in between the two cells. And then these arrows here represent what those lytic granules are releasing onto the target cell. Again, ideally to cause apoptosis rather than lysing the cell, which can potentially release whatever has infected this cell to go out and infect other cells in the body. But a dead infected cell is better than a living infected cell, so if apoptosis doesn't work, lysis is better than nothing. And then you'll have cells in the vicinity that help fight off other cells that get infected by the same thing. The last part of the activation stuff here is about the B cells. B cells, when they're naive, when they're mature but not yet exposed to their actual antigen, have on their surface antibodies that kind of look like receptors. They haven't been secreted to float around separately. They're still attached to the cell. And so this B cell is waiting around in your lymph nodes or whatever, waiting to finally be introduced to the antigen that it's particular for. When it is, that antigen binding will sensitize the B cell. And a sensitized B cell will show part of that antigen on its surface. Now, in this picture, the steps that they're showing here between antigen binding and sensitizing the B cell leaves a lot out. What happens actually is the B cell that has that antibody that's attached to the antigen pulls the antigen inside. The antibody acts as a receptor so that it can be pulled inside, and then it presents that on its outside surface, like an antigen presenting cell, but this isn't technically an antigen presenting cell. But it does that so that the sensitized B cell can interact with the helper T cell. T cells get a co-stimulating factor from the antigen presenting cell to be activated. But the B cells only interact with the antigen directly by itself. There's no antigen presenting cell showing the B cell the antigen. So the B cell has to show the antigen to the helper T cell, and the B cell is presenting that antigen on its surface with class 2 MHC, and the T cell receptor recognizes the antigen. The CD4 protein on the helper T cell will recognize class 2 MHC, and that will cause co-stimulation. The cytokines that helper T cells release help to activate these cells. With the cytotoxic T cells, they help when necessary, when the antigen presenting cell can't co-stimulate itself. But for B cells, helper T cells are necessary for activation. Because without it, then you just have sensitized B cells that are not going to fight off an infection. They have to be fully activated with the help of the helper T cell. Hence, that's why they're called helper T cells. What they're releasing belongs to the group called cytokines. Specifically, they're releasing a molecule called interleukin. Inter meaning between, and leukin referring to between white blood cells. Interleukin is abbreviated IL, and there's a whole bunch of interleukin molecules. The one that's specifically used for helper T cells to activate B cells is interleukin 2, or IL-2, you'll see sometimes. When they're exposed to IL-2, the sensitized B cells will then become activated, proliferate, and make a bunch of copies of themselves that all make the exact same antibody against the exact same antigen. And they'll differentiate. Some of them will become memory B cells for future infections of the same thing. And the other ones are activated B cells, which further differentiate or mature to become what we call plasma cells. Now in the picture here, the difference between the activated B cell and the plasma cell is what's in the cytoplasm. The plasma cells develop an extensive intramembranous system, which is basically rough ER and Golgi apparatus, which are necessary to secrete the proteins that are antibodies. Okay, so all of this stuff in the cytoplasm here is just so that plasma cells can start secreting lots and lots of proteins, lots and lots of antibodies. So that brings us to antibodies, which I've actually kind of already talked about a little bit, so I'm going to do this quickly, or I'm going to go through a couple of these things quickly because I already talked about it. But here's the basic structure of an antibody. I showed you one earlier. There's a light chain and a heavy chain. There's two of each of those, making a Y chain. The outer end of the light chain and the heavy chain are variable segments, which is where the DNA gets randomized to make a specific, to make a unique antibody from that lymphocyte. After the DNA is randomized at the beginning of the lymphocyte's life, it never changes, and so it always makes the exact same antibody with the exact same antigen binding site. And antibodies have two antigen binding sites. Then the other half of the light chain and the corresponding part of the heavy chain, there's a constant region there, and then the rest of the heavy chain, the two of which then meet up to make sort of the body or the stem or whatever the antibody. There are two more constant regions or segments, that's the word I was looking for earlier. The two constant regions in the body or stem of the antibody contain in one of them a complement binding site, which is how the complement protein 1 can bind to antibodies for the classical pathway for complement. And there's also a spot where they can bind to, it says macrophage here, but it'd be more appropriate to just say phagocyte. So when an antibody is bound to the surface of an antigen, that can cause phagocytes to be able to ingest that thing a little bit easier, which is what we call opsonization. Now this next picture I'm going to go by real quick because it's out of another book. Whoops. Didn't go where it was supposed to. There we go. And I prefer this picture to show you the real structure of antibodies. This is from the Protein Data Bank website. It's just a really nice representation of the actual structure of antibodies. all of the little round parts that we see here represent specific atoms in the structure of the protein. The yellow parts are the light chains, and then the red and orange parts are the heavy chains. And on this side over here, we can see sort of a cluster of atoms, which is the variable region of the heavy chain here, and the variable region of the light chain. Then you'd have the next loop, which is constant regions of both of them, and then a constant region here and a constant region here down the heavy chain. Just corresponding to the schematic picture I was showing a second ago with the loops, you can kind of see the basic structure there. I've always found this picture kind of amusing. To me, it's sort of like a mutant sesame bagel. But the point of this is to suggest how antibodies actually interact with the antigen. Because of the specific unique structure of the antigen binding site, it recognizes a particular molecular signature, if you will, of the antigen. And those spots that it recognizes are called antigenic determining sites, or simply epitopes, which are sort of the sesame seeds here. And so when this antigen gets into your body, then the antibodies that recognize that specific epitope can bind to it. Now let's pretend that this isn't a sesame bagel, but let's pretend it's an everything bagel. So it has sesame seeds on it, and it has poppy seeds on it, and it has dried onion flakes on it, and I can't think of what else you'd have on the surface of the bagel. Maybe a little oat, dried oat things, whatever. So each of those different things would represent a different epitope. So you can have different antibodies that all recognize the same antigen, because antigens will have a multitude of epitopes on them. This particular example is just, they didn't draw to suggest a sesame seed bagel necessarily, but that's just what I get out of it. So there are spots specifically on the thing that the antibody binds to. Now, what the antibodies do when they bind to the antigen is listed here. Our book doesn't present this as a list exactly, but I like this list because you can remember it based on this mnemonic. Now, when I started teaching, I used a book that presented this information this way, essentially, but they only showed the first four things. The mnemonic was planned. I added on the end of this opsonization, so Plano is the mnemonic, because I think it's important to recognize that there is this fifth thing that can happen. So P stands for precipitation, which is what happens with small antigens, which would be molecules. And antibodies can bind to them. One antibody can bind to two of them, and then one of them can be bound to multiple antibodies, and it can form a big cluster of antigens and antibodies all together. Small antigens are probably dissolved in your bodily fluids, and as they cluster together into bigger and bigger groups, they become less likely to dissolve into the fluid, and they precipitate out. Then they can be cleared away by phagocytes because the antibodies attached to them are opsonizing the antigen also. Lysis just refers to complement. It doesn't say complement or cytolysis here because we want an L in that spot for the mnemonic. But the classical complement pathway is started by antibodies binding to the thing. So let me just jump back to this picture for a second. So let's say this is a cellular antigen. There's a complement binding site on the antibody here, and so complement protein 1 will come join this and get complement proteins 2 and 4 involved in breaking down complement protein 3, which will recruit complement proteins 5, 6, 7, 8, 9 to form the membrane attack complex and make a hole of the antigen, which we talked about before. But that'll lead to lysis. Okay. Agglutination is very similar to precipitation, but it's for large antigens, which means cellular antigens. You should recall the word agglutination because it came up when we were talking about blood typing. When you are typing blood, you're exposing blood to antibodies against the A antigen or the B antigen or the Rh factor antigen to see which antigen is present on the blood. And when you get a reaction in the blood typing reaction test, you call that agglutination, that clumping of the red blood cells together in the vial or on the microscope slide that you're doing the test on, whatever. But any cellular antigen, that will happen to. They'll be clumped together because they'll be cross-linked by multiple antibodies. Neutralization is when you make it impossible for the antigen to do what it's supposed to do. The best example of what that would be is for like a viral particle. Let me go back to this picture again, just because it's convenient. Let's say these epitopes here represent the part of the virus that can actually bind to the host cell and cause infection. If the antibodies bind to the epitopes, they block the ability of this thing to infect its intended target. Even if this Y here at the bottom is blocking those two, the exposed ones aren't necessarily going to be able to cause infection either because the thickness or the height of the antibody interferes with the ability of the virus to actually get up next to the target cell. So it neutralizes the virus because it can't actually cause infection anymore. You can neutralize other types of things, bacteria that have cilia or flagella for movement. If the antibody binds to that structure, then the cell can't move, and that can neutralize them too. And then opsonization by itself is possible, which is because antibodies have a binding site for macrophages and phagocytes. When something binds to it, a white blood cell can just come along and bind and destroy it. Now, I added this onto the end because that can be all that happens. But really, opsonization is the endpoint of all the other four. So when you precipitate or you lyse or you agglutinate or you neutralize an antigen, because the antibodies are attached to it, that means that it will be cleared away by phagocytes, opsonized. Opsonization is involved in all those other ones, but it can be the only thing that happens too. So there are a few other things to cover about antibodies to close out the immune system stuff. Obviously, there's not time to today. So we'll pick up with this on Tuesday. In the first 10 minutes of class, we'll finish off the immune system, and then we'll definitely start the respiratory system next week. So we'll call it a day there and wrap this up next time.
Do we have lab next week?